time because novel and more efficient anti-obesity strategies are being developed, and that is thanks to our increased understanding of the underpinnings of hyperphagic obesity. Some of the compelling things that we've learned in the recent years is that obesity is largely explained by genetic variants, some of which can make people feel constantly hungry, as if they had been starved for 3 days. And most of the genes that contribute to obesity are expressed and active in the brain, which the mammalian brain really, until recently, remained a big black box. And more specifically, we found that these genes are involved in nutrient and metabolic sensing, as well as in brain connectivity and plasticity. And there is currently a huge effort to map neuronal circuits involved in appetite control. Factors that determine decision-making when food is available include, of course, homeostatic needs, so the needs of the body to process and do its normal functions, but also hedonic drives for foods that are palatable, sensory inputs, and as well psychological factors like mood and stress. And historically, people really focused on the characterization of the processing and integration of homeostatic needs by the brain. But in fact, really, most people focus on energy needs. But we know that homeostatic drives that make us eat are not just determined by the lack of calories, but also by the need to obtain a balanced diet to meet our physiological needs. In fact, we know that across species, from worms to humans, specific appetite for key nutrients, including the 3 macronutrients, protein, carbohydrate, and fat, but also for sodium and calcium, exist. And these appetites are universal, and they have evolved to guide animals to obtain specific nutrients and help animals balance their diets. In an environment where these key nutrients are available in a balanced manner, macronutrient-specific appetites may not play a key role in driving food intake. However, when one of these nutrients becomes limiting, the specific appetite for these nutrients becomes the main determinant of feeding behavior, often to the detriment of energy balance. So we can ask ourselves, are our modern diets balanced? And if they're not balanced, which is the macronutrient that is missing? And are there some specific macronutrient appetites that drive feeding behavior in the modern environment? So one of the macronutrient-specific appetites that seems to be a strong regulator of feeding is the appetite for protein appetite. And maybe that's because there is no tissue storage for protein in the mammalian body, unlike for carbohydrate and fat. And that may explain why protein is a macronutrient that has the strongest impact on feeding behavior. And this notion is well illustrated with this graph that summarizes the finding of a meta-analysis where they gather the nutritional data from 68 experimental trials in which participants had ad libitum access to food with different proportion of protein. And what is very striking is that participants obtained diets or selected diets with different concentration of protein, from 8% of energy as protein all the way up to 55%. But in spite of this wide range of protein concentration in the diet, absolute protein intake remained relatively stable. And in contrast, energy intake varied massively. So one interpretation is that there are mechanisms in place to ensure that the protein intake is stable and is regulated at the expense of energy intake. When humans or animals are on a high protein diet, they eat less because as soon as they reached the sufficient amount of protein, they're sated. Whereas when animals or humans are eating a low protein diet, there are mechanisms that push them to eat more and more to reach this target protein level. So this data maybe suggests that protein intake is tightly regulated, and this control may in fact be prioritized under certain conditions over the control of carbohydrate, fat, and energy intake. So this is quite compelling, I think, to support the idea that protein intake is regulated and there is some kind of proteostatic control of energy intake that can become relevant under specific circumstances. In the modern adipogenic environment, we know that protein is getting diluted. And we know that, for example, from this NACE data, so that's nutritional data from the United States that were collected over the years. And we know that in the 70s, the typical diet contained roughly 70% of protein. And in the years 2000, 30 years later, they measured a decrease in the concentration of protein in the diet. So we know that in the same period of time, obesity prevalence increased. This was associated with an increase in caloric intake, so that's okay. But what's quite remarkable is that in the same time frame, daily protein intake remains stable. So perhaps the proteostatic control of energy intake could be a major driver of the current obesity epidemic. This is the protein leverage hypothesis that has been developed by Steve Simpson and David Oppenheimer. But so if that's true, maybe targeting these mechanisms may provide new strategies to address the obesity epidemic. So this brings me to the key questions that my lab is investigating. How does a mammalian brain sense protein availability to create representations that regulate appetite? The model of choice to investigate this question is the mouse, because of the modern neuroscience toolbox available to study mouse neural circuits, and it's a good model to understand how the human brain works. However, data supporting the protein leverage hypothesis in the mouse are controversial. And that's not really the focus of my talk today, although I'm happy to take any question on that. But however, I'd like to begin my presentation by sharing some data that we have produced that do support, at least in part, this model in the mouse. So I have shown you this graph before, supporting the conclusion that protein intake may be tightly regulated when humans can select the dietary composition, the macronutrient composition of their diet. So I think this graph is quite compelling to support the idea that protein intake is regulated. So we wondered if we could produce similar type of evidence in mice. And that's not necessarily straightforward because the macronutrient composition of laboratory diets is fixed. And when we put mice on a self-selection paradigm where they can choose themselves the macronutrient composition of their diet, all animals roughly select the same diet with the same amount of energy, the same amount of protein in the diet. So we can't really have something like that. But we thought about the fact that mice in laboratories are housed at 24 degrees Celsius, which is not an ideal temperature for them. Their temperature of thermoneutrality is roughly 28-30 degrees. What does that mean? It means that at 28 degrees, the animals don't need to produce heat to maintain their body temperature. So when we house them at 24 degrees, they need to produce heat to maintain their body temperature, and this increases their energy expenditure. But remarkably, if you house mice at different ambient temperature, they will increase their energy intake to meet these higher energetic needs to maintain body temperature. So based on that, we thought that maybe if we house animals at lower temperature and look at how they would self-select their diet, we may learn something about the regulation of protein intake. So what we did is we starting by housing mice at 18 degrees, and throughout the experiment, the animals could self-select the macronutrient composition of their diet. And then we increase the ambient temperature gradually. So during this gradient, energetic needs decreased because the energetic demand decreased. And long story short, we were able to produce this graph showing, so first of all, that we obtain conditions where caloric intake varied massively. So at lower temperature, animals ate more calories. At thermoneutrality, they ate less calories. And these changes in energy intake were produced essentially by changes in carbohydrate intake. And in these very different conditions of temperature and of energetic intake, protein intake remains stable. So we think this is some piece of evidence to suggest that there are some mechanisms that maintain protein intake stable independently of energy intake. So that's quite important for us. So how does the brain sense proteins, the proteins in the body? We know that when proteins are digested, This leads to the secretion of gut-derived signals like CCK and GLP-1 that act on local afferents that project to the nucleus of the solitary tract in the hindbrain. And this is a region very important for the control of food intake. And then, of course, proteins are being digested, and then this leads to the release of amino acids that are metabolized locally and by the liver mostly. But there is some variation of circulating levels. But very importantly, the liver is a key organ of amino acid detection, and but the adipose tissue as well. And what happens after a protein load is that other signals that regulate food intake are being modulated. And these include leptin, insulin, but also FGF21. And these signals are being detected by key regions in the brain that regulate appetite, including the mediobasal hypothalamus. So this is well characterized, and this likely contributes to the way proteins are sensed and modulate appetite. However, one problem I have with this is that none of these signals are really specific for proteins. These signals are also modulated by other macronutrients. So it's difficult to understand how these signals may mediate the protein-specific appetite I've just explained about. So we think that there must be something else that specifically encodes protein availability. So how is protein availability encoded? So based on the literature, and so I can't explain how I made this list, but basically I made a list of how an ideal signal of protein availability should look like. So we think it should have circulating levels reflecting dietary protein intake. It should be specific to proteins and independent of carbohydrate and fat and energy. It should produce rapid changes in protein intake and protein metabolism because there is no storage compartment for protein, so there is no buffer to allow a transition. So it should be quite rapid. But also, we know that it should inform on the anabolic value of the protein load. And last, this signal should be detected by the brain, and more specifically by neural circuits in the brain that regulate appetite. And we propose that circulating levels of the branched-chain amino acid leucine is a good candidate to encode protein availability. So it ticks all the boxes. I'm not going to show the data. And so what we're doing in my lab is use brain leucine sensing as an entry point to characterize protein sensing pathways. So after the injection of a protein meal or high protein diet, one of the few amino acids that is changing in the circulation significantly and not completely metabolized by the liver and the intestine is leucine. And we are looking at the idea that leucine and maybe other amino acids are detected directly by specialized neurons in the brain that control appetite and metabolism. So this is our working model. In response to the ingestion of a high protein meal, there are changes in circulating level of leucine that are detected by specialized neurons in the brain that are leucine sensing neurons that we think may be enriched in metabolic sensing areas of the brain, like the mediobasal hypothalamus or the nucleus of the solitary tract. And when these neurons sense leucine, this modulates their electrical and synaptic activity, leading to the modulation of downstream circuits that regulate appetite, body weight, and eventually metabolic health. So we've been looking at this and trying to answer several questions. The first one was, what are the behavioral consequences of central leucine detection? And does it reproduce what we would expect in response to a high protein diet? And then the next question was, who are these, if they exist, who are these leucine sensing neurons? Can we characterize and identify them? And the last question was, can we target leucine sensing neurons to modulate energy balance? So to model the situation of increased circulating leucine levels during a high protein meal, we've used the following paradigms. We've used rodents, and most recently mostly mice, that were fasting during the day, which is their inactive phase. And they are fasting during the second half of the day to produce a mild fast. And then we injected them with leucine in small regions of the brain that are enriched in nutrient sensing neurons. And we injected them 1 hour before dark because this is the moment of the day where the animals are most likely to eat a big meal. They are really hungry after a fast and just before dark onset. And we injected them with nano amounts of leucine to mimic physiological increase in brain leucine concentrations. And this remains a pharmacological intervention, but we're trying to be as physiological as we can. And what we found is that leucine suppresses food intake across 3 different timescales, which is quite interesting. So like I said, these animals are fasted, so they're all quite hungry. And the control animals in white here, when we give them a food access after the injection, they initiate the first meal quite rapidly. But in contrast, when the animals are injected with leucine, the first meal, the initiation of the first meal is significantly delayed. Leucine also reduced meal size and on a longer time scale reduced meal number. So we found that leucine can actually modulate different aspects of feeding behavior across different timescales, and we think this is happening via the modulation of different neural circuits that we wanted to characterize. Importantly, these effects are not associated with the production of conditioned taste aversion, so this is not a sickness response. So in the subsequent study, we've used this paradigm to characterize what is going on. Again, this is our model. I'm not going to go over this again. So what we did first was develop a model to characterize and identify leucine-sensing neurons. We choose to use primary cultures of adult hypothalamic neurons, and by adult, I mean neurons that are developed in culture. So they're collected from mice that are 6 to 7 weeks old, so they're definitely post-weaning, maybe adolescent. But and we maintain these neurons in a media that contains nutrients in concentration similar to what is found in the brain. So we reduced the concentration quite dramatically. But what was quite nice is that when we use calcium imaging and expose these cultures to leptin and glucose, we find an expected proportion of neurons that respond to leptin and glucose. And by expected, I mean the fraction of neurons responding to leptin and glucose is the same as what you would find with electrophysiology or more traditional techniques. So then when we expose these neurons to leucine, we found that some neurons were activated, some neurons were inhibited. So leucine was able to produce both activation and inhibition, but the majority of them were not responding. So this is not a ubiquitous non-specific response. It seems like some neurons have the ability to respond. And when we use culture developed from transgenic animals expressing reporters in specific neuronal populations known to be involved in appetite control, like POMC neurons or MPY neurons, you may have heard about these neurons that are key neurons that control appetite, we found that a fraction of each of these types also responded to leucine. So it seems like leucine can produce different kinds of response in some neurons only, but these neurons are molecularly heterogeneous. And in total, roughly 20% of the hypothalamic neurons responded. Only a fraction of them were identified with our reporter lines, and the majority of the leucine-sensing neurons remained unidentified. So to identify these neurons, we've used a technique called phosphotrap, which is based on the observation that in activated neurons that express the classical marker CFOS, ribosomes are phosphorylated. And we can use immunoprecipitation to pull down ribosomes of activated neurons. And then, using differential transcriptome analysis, we can obtain a molecular signature of leucine-activated and leucine-inhibited neurons. So we've done that, and it gives a lot of data, maybe more questions than answers. So we decided to combine this with another approach that was quite complementary, in which we used our primary culture model again, and we use calcium imaging to identify leucine-sensing neurons or leucine non-responsive neurons. And then we collected the intracellular content with a pipette, with a micropipette under the microscope. And then we performed single-cell RNA sequencing of cells that were either activated, inhibited, or non-responsive to leucine. And again, this gave us a molecular signature. So when we cross these databases, we obtain top candidates. It's still giving us a lot of questions, maybe more than answers, but there are some candidates that we thought were worth investigating further. And one of them is Carcitonin Receptor. So Carcitonin Receptor is expressed in many areas in the brain, including in nuclei that regulate feeding. It's the hypothalamus and the brainstem, the two areas I've talked about before. And we know that agonists against calcitonin receptor decrease food intake and are used in novel anti-obesity therapies under development. So we thought that these neurons may represent a good candidate population mediating leucine and proteins anorectic action. So we first confirmed that leucine can indeed activate these neurons with an actual experiment. And what we did is deliver leucine locally into the nucleus of the solitary tract in the same manner as the one I've described before. So we give a local small injection of leucine, and then we process the brain tissues for multiplex in situ hybridization with RNA-scope. So with this technique, we can label calcitonin receptor expressing neurons in the nucleus of the solitary tract. It's also expressed here in the AP. But when we look at tissues of animals injected with leucine, we see FOS, which is a marker of neural activation, that is increased. So increased FOS expression, which we knew about before. So leucine activates some neurons, unlike the vehicle injection. And what we see is that a number of these FOS-positive neurons are also calcitonin receptor positive. So while there are lots of purple neurons that are not green, the vast majority of calcitonin receptor expressing neurons, the green ones, are also purple. So in short, leucine activates the vast majority of calcitonin receptor expressing neurons in the nucleus of the solitary tract. What we did next is test the role of this activation in leucine-induced anorexia. So we used a strategy to specifically silence calcitonin receptor expressing neurons in this area of the brain in adults. So this was using a viral technology. And we measured the anorectic response to leucine in these animals. So we have the control animals that receive a control virus, and when we give them leucine into this area of the brain, we have the expected anorectic response. However, when we silence these neurons specifically, the response is blunted and almost disappeared, directly involving activation of these neurons in leucine sensing in this area of the brain and in leucine's anorectic action. So something I didn't mention before is that when we give leucine into the hindbrain, into the nucleus of the solitary tract, we see that leucine delays myelination in fasted mice. So this graph I've shown you. But this is in fact quite remarkable because this area of the brain is not classically associated with the regulation of myelination sites. So this suggested to us that because the regulation, sorry, let me start again. So this was intriguing because the nucleus of the solitary tract is classically associated with the regulation of meal size and not the regulation of meal initiation. Meal initiation is typically believed to be the job of AGRP neurons in the hypothalamus. So this suggested to us that nucleus of the solitary tract neurons here that can sense leucine maybe communicate with AGRP neurons in the hypothalamus, whose job is to regulate meal initiation. So to interrogate the potential connectivity between AGRP neurons and neurons in the nucleus of the solitary tract that sense leucine, we use a queue-dependent polysynaptic retrograde tracing strategy using a virus called PRV introvert, developed by Jeff Friedman. And we gave this virus into the mediobasal hypothalamus, the arcuate nucleus here, of AGRP iris queue mice. So when the virus arrives into AGRP neurons that have queue, the virus can start to replicate and propagate backwards, and it jumps the synapses, and it can label all the neuronal inputs that go and project to AGRP neurons. And then there is this HA reporter that is expressed, so we can visualize all the inputs onto the AGRP neurons. So with this technique, we labeled all the areas that had been previously shown to project to AGRP neurons. But interestingly, we also found some labeling in the nucleus of the solitary tract, including in cells that also express calcitonin receptor, providing direct evidence of a neuroanatomical connection between NTS calcitonin receptor neurons and AGRP neurons. So then we wanted to determine whether this circuit was important for leucines and proteins anorectic action. So we've characterized the circuits first, and I'm not going to get into the details of how we've done that, but basically we found that leucine can be sensed here by these calcitonin receptor expressing neurons that project to the dorsal major hypothalamus, and these neurons expressing leptin receptor, which in turn inhibit LGRP neurons. So LGRP neurons normally produce myelination, but when they're inhibited after leucine sensing in the hindbrain, this blocks and delays myelination. So to test the relevance of the circuit in the anoretic response to leucine, we use another viral approach to silence neurons, but this time we silence neurons in the DMH here that are activated by leucine sensing in the NTS and project to LGRP neurons. So we silence these neurons and we characterize the feeding suppressive responses to either NTS leucine injection here or DVC, it's the same area. So we looked at the effect of leucine when it's injected here, and we also looked at the anorectic response to a high protein diet. So first, when we gave leucine into the hindbrain here and looked at the anorectic response to leucine in control animals, we measured the expected decrease in food intake. However, when this portion of the circuit here is silenced, animals no longer suppress their food intake in response to hindbrain leucine injections. And this was also associated with a decrease in the body weight suppressive action of hindbrain leucine sensing. And then we use another paradigm where animals were maintained on a standard diet containing 20% of protein. And then we switched them to a high protein diet where protein represented 45% of the energy. So control animals, when we do that, they have a deep decrease in food intake upon introduction of the high protein diet. Then this goes back up a little bit, but still remains significantly lower than in the baseline condition. So normal animals would eat less in response to a high protein diet, and this is associated with a decrease in body weight. And when we silence this portion of the circuit, we notice that the hypophagic response to the high protein diet was blunted. And then we also measured that the body weight suppressive effect of the high protein diet was completely gone. So these data support a role for calcitonin receptor expressing neurons in the nucleus of the solitary tract in the response to brain leucine sensing, but also in the response to high protein diets. Another candidate that came up with our strategy to characterize the molecular signature of leucine sensing neurons is CAC91G. So CAC91G is also significantly enriched in leucine sensing neurons and is known to be involved in the regulation of the excitability of some hypothalamic neurons, including POMC neurons. So the reason we decided to further investigate this particular candidate is that there is a literature showing that CAC91G, which regulates theta calcium channels, is involved in the response to high-fat diet. And we know that animals that don't have any CAC91G whole body knockout animals are protected against diet-induced obesity. And also we know that systemic pharmacological inhibition of CAC91G protects against diet-induced obesity. But the underlying mechanisms are not known. So we thought this was worth investigating. In the hypothalamus, CAC91G is particularly enriched in the arcuate nucleus of the hypothalamus. Like I said, we know that some POMC neurons express CAC91G. But when we looked at existing transcriptomic data of this area of the brain and all the neurons, it shows that CAC91G is expressed in many different neuronal types, including POMC neurons, but theta neurons, TAC2 neurons. So it's not really straightforward. It's a very heterogeneous population. In primary culture, so we use the same model with primary culture with calcium imaging. We found that pharmacological inhibition of CAC91G completely blunts leucine-induced rapid changes in calcium concentrations. So CAC91G somehow is required for leucine-induced activation of these neurons here. And in vivo, when we pre-treated mice with a local injection of the same inhibitor, we found that inhibiting CAC91G blunted leucine-induced anorexia in mice. And these were given in the medial basal hypothalamus because this is where we identified this gene. So giving us some evidence suggesting that Cac9-1G expressing neurons could be leucine sensing neurons. So now we're trying to take this further, and we're developing a viral approach to CRISPR knockout Cac9-1G specifically in the arcuate nucleus of the hypothalamus. So I'm not going to show everything we have, but basically there is no phenotype when the animals are maintained on a normal diet or on a high-fat diet. So at least this tells us that whatever happens in whole body Cac9-1G knockout animals, it seems like it's not because of the medial basal hypothalamus, because when we remove Cac9-1G from the medial basal hypothalamus, the animals respond to diet-induced obesity in the same manner, to the same extent. So, however, when we gave these animals a local injection of leucine into the hypothalamus, we found that the anorectic response to leucine in the hypothalamus was blunted, suggesting again that neurons expressing T-type calcium channels, so this is Cac9-1G subunit, may be leucine sensing neurons. So then we fed these animals a high protein diet, again the same diet, a high protein diet containing 45% of the P45 diet, so 45% of protein in the diet. And what we found is that in comparison to the controls, the anorectic response to the high protein diet was blunted, and this was also associated with a decrease in the body weight suppressive effects of the high protein diet. So we're still actively investigating the underlying mechanisms, and in particular, we're trying to test whether leucine directly engages these channels, because this could be just a marker of leucine sensing neurons, and when we remove it, it just perturbates these neurons. But we think maybe leucine may be directly changing the activity of these channels. So that's something we're looking at. So in the, so that's just the summary of the findings so far. We think that leucine is detected by a distributed network of neurons in the brain, in the hindbrain, and in the hypothalamus, and regulates these different distinct behaviors that collectively contribute to the anorectic response to leucine, both meal initiation, but also meal size and meal frequency. And we think these different behaviors are controlled by different circuits and by different types of leucine sensing neurons that include calcitonin receptor expressing neurons, also POMC and ARGRP neurons, but also neurons expressing Cac9-1G. So now I'm going to, in the next 10 minutes, I'm going to talk about a new research axis in my lab on nutrient sensing in the median eminence. So that's a bit of a shift, but I think it's worth it to introduce this today. So the median eminence is a tiny structure here at the base of the hypothalamus. And it is mostly made of non-neuronal cells that we would call glia, but it really doesn't mean much. So non-neuronal cells include astrocytes, oligodendrocytes, tanocytes, microglia. So it's mostly non-neuronal cells here. And the median eminence has gained momentum recently because it's highly plastic and because of the role of tanocytes here in nutrient sensing. Tanocytes are the cell types that line the ventral wall of the ventricle above the median eminence. And these cells have been shown to be highly nutrient sensitive, highly plastic, and play an important role in the regulation of energy balance. The median eminence is often considered as the bidirectional gateway between the central and peripheral endocrine system. It is one of the circumventricular organs which has an open blood-brain barrier. And it is a place where hormone-releasing neurons from the hypothalamus terminate and release the hormones that regulate many functions, including growth, reproduction, stress, and metabolism. And because of its exclusive access to circulating factors, it is also exposed to signals that do not normally enter the brain or do not enter the brain to the same magnitude. And so signals from the circulation access this territory, therefore what happens is that nutrient and metabolic sensing neurons in the arcuate nucleus send a projecting dendrites into the median eminence to have a test of what's going on in the periphery and to have a representation of peripheral energy and nutrient availability. But what we know is that there is a diffusion barrier between the median eminence and the arcuate nucleus to replace the missing blood-brain barrier, because this area is quite toxic, in fact, for neurons, very sensitive cell types. It's probably glucotoxic, lipotoxic, so cell bodies should not sit here, neuronal cell bodies, because it's too toxic for them, but they still want to know what's going on in this area. And so this diffusion barrier can be shown here when we inject in the periphery a blood-brain barrier impermeable dye, Evans blue, and we see that this dye that does not cross the blood-brain barrier still will be present in this territory of the median eminence and a little bit in the base of the arcuate nucleus of the hypothalamus. So that's the median eminence, a very interesting nutrient sensing area. And one of the reasons we became so interested in the median eminence is because we were interested in mTOR. mTOR is a well-known nutrient and protein sensing signaling pathway, and we've been looking at the role of mTOR in the regulation of appetite for a while. But when we stand against phospho-mTOR, which is the active form of mTOR, what we found that was quite remarkable is that phospho-mTOR is extremely active, expressed in, so mTOR is extremely active in a population of cells here that sit between the median eminence and the arcuate nucleus of the hypothalamus to a level that is not seen anywhere else in the brain. So there are some cells that have super active mTOR in the median eminence, and we wanted to know who these cells were and what this meant. And what we found is that all these cells are actually oligodendrocytes. They're not anything else. They're not neurons, not astrocytes. They're all oligodendrocytes, and the expression of phospho-mTOR in these cells is regulated by nutritional signals that we mimic with fasting and refeeding. So when energy is available, mTOR becomes active in these cells, and it's also regulated by the amount of protein in the diet. So there is a population, in short, of oligodendrocytes here that express phospho-mTOR, and this expression is regulated by the nutritional content of the diet, and so protein and energy availability. So that may be a new protein sensing populations that we are not aware of. And what this revealed to us, at least, that we were not aware of, is that there is a population of oligodendrocytes here. We didn't know anything about them. And what's quite striking that you can see in this video, if I can start it, where we stand against MBP, which is a myelin-specific protein, is that myelin is absent from the rest of the hypothalamus because it's gray matter, but highly present in this band that sits between the median eminence and the arcuate nucleus of the hypothalamus. So we know that mTOR is a major regulator of myelination, because mTOR regulates many anabolic processes. And we know that when myelin is made, it comes some oligodendrocytes come from oligodendrocyte progenitor cells that can become newly formed oligodendrocyte during differentiation of these cells, and then mTOR becomes active during myelination. And mTOR, of course, integrates growth factor signals and nutrient and energy-related signals, and it is known to integrate these signals in the control of myelination, which involves lipid biosynthesis, protein translation, cytoskeleton reorganization. All these functions are not known to be controlled by mTOR. But it's quite surprising to find active mTOR in the adult median eminence, because oligodendrocytes are normally mostly produced before adolescence. And the image I showed you before was taken for a 12-week-old mouse. So the idea that there is active myelination in the adult brain is a bit unexpected. So normally oligodendrocytes, like I said, are produced before adolescence and are long-lived. Progenitors, OPCs, can be recruited during myelin repair. And there is, however, an emerging literature on adaptive myelination that can occur during adulthood in response to physiological cues, like, for example, the acquisition of a new motor skill. But this myelin adds to existing myelin. So all of this together raised many questions regarding the role of active mTOR signaling in the adult median eminence and its links to nutritional signals. So what we did to look at this is use single-cell RNA sequencing of microdissected mouse median eminence samples. And one of the things we found was that in this area of the brain, oligodendrocyte lineage cells are present in three states: the progenitors, the myelinated adult oligodendrocyte, but also a significant proportion of newly formed oligodendrocytes that are characterized by the expression of BMP4. So we confirmed this with our anescope, and indeed we found that there was a significant population of cells expressing BMP4 in the oligodendrocyte lineage. So I'm not going into the details of this, but this confirms that there is newly formed oligodendrocytes, and they represent 25% of the total population, which is really, really high. So we then tested whether the production of oligodendrocytes may be sensitive to nutritional signals, and we used a fasting refeeding paradigm. So in the fasted state, energy is lacking. In the refeeding state, energy is available. And this is a classical paradigm to study nutrient sensing pathways. And what we found when we did that and coupled with single-cell RNA sequencing is that oligodendrocyte lineage progression is one of the top pathways regulated by nutritional signals. And this was confirmed again with the RNA-scope. So this is a complicated slide, but we stand for stage-specific markers in the median eminence in animals that were fasted or refed or ad libitum fed. And what we found is that refeeding significantly increased the expression of markers that are specific to newly myelinating oligodendrocytes and also the number of new oligodendrocytes. So this experiment told us that nutritional signals, what they do to oligodendrocyte lineage cells is increased differentiation. But whether that means increased myelination, that we didn't have any evidence of that. So I'm going to skip that because I think I'm going to talk too much otherwise. So, but what we found, so this is a schematic to summarize what we have found so far, is that there is a population of oligodendrocytes that are myelinating at the border between the median eminence and the arcuate nucleus of the solitary tract, and the arcuate nucleus of the hypothalamus. And this population is plastic because there is continuous production of new oligodendrocytes, and production of new oligodendrocytes is actually regulated by nutritional signals like refeeding and, like I said before, like protein. I'm going to go a bit quickly over this, but we wanted to identify potential mechanisms through which oligodendrocyte plasticity in the median eminence may influence the function of local neurons that are critical to the control of appetite. And when we looked at our single-cell RNA sequencing data, what we found is that many of the proteins associated with the formation of the extracellular matrix are enriched in oligodendrocytes and nutritionally regulated. So I'm now going to introduce perineuronal nets, which is a structure, an extracellular matrix structure that has been known to be involved in many processes during development. But recently, the laboratory of Michael Schwartz describes the presence of these extracellular matrix structures in the adult arcuate nucleus of the hypothalamus. So these perineuronal nets can be visualized with an immunostaining against WFA, so that this basically labels the GAG chains of these ECM extracellular matrix structures. So we can visualize these perineuronal nets. And many of the components are actually expressed specifically by newly formed oligodendrocytes and myelinating oligodendrocytes. So these perineuronal nets are normally formed during developmental maturation of the hypothalamus, and they regulate synaptic input onto the arcuate neurons, as well as local diffusion properties. And so that's an attractive candidate mechanism through which oligodendrocyte may regulate arcuate nutrient sensing. So to look further into this, we stained against perineuronal nets using the same staining in the median eminence of fasted and refed animals. And what we found is that refeeding leads to a significant increase in this staining that labels perineuronal nets, suggesting that there is some kind of remodeling of the local extracellular matrix during the fasting refeeding transition. And one of the top genes differentially expressed in oligodendrocytes during the fasting refeeding transition is ADAMS TS4, which is an enzyme, a metalloprotease, involved in ECM degradation. And what we found is that ADAMS TS4 is specifically expressed in myelinating oligodendrocytes that are co-labeled with either BMP4 or labeled here with SOX10. So Adams TS4 is enriched in oligodendrocyte lineage cells here. And during the fasting to refeeding transition, there is significant decrease in the expression of Adams TS4 in oligodendrocytes. So this could mediate the nutritional regulation of perineural nets. So to confirm the role of oligodendrocyte plasticity in the regulation of the local extracellular matrix in the median eminence, we looked at the density of perineural nets in MRF knockout mice. So MRF is a transcription factor that is required for the differentiation of oligodendrocyte progenitors. So in these mice lacking MRF in adulthood, it's an induced model, the formation of new oligodendrocyte is blunted. And what we found is that these mice lack perineural nets in the median eminence. So that's median eminence specific. And so this directly implicates adult oligodendrocyte production in the formation and the maintenance of the local extracellular matrix. And so last, we assessed the functional consequences of PNN disruption in the accurate median eminence region using injections of congruitinase ABC, which is showed here. So, and this is an enzyme that can digest the extracellular matrix dark chains effectively, and it disassembles perineural net scaffolds. So this resulted in an increase in weight gain in the animals that received the enzyme compared to control, and an increase in food intake. So these results support a role for median eminence oligodendrocyte plasticity in perineural net remodeling, which seems to be important for the regulation of energy balance. And so that's sort of a summary working model that we have. We've shown that there is a population of myelinating oligodendrocytes here that are quite plastic, and we think that these oligodendrocytes regulate the local extracellular matrix, but also may be involved in the regulation of the permeability barrier between the median eminence and the arcuate nucleus of the hypothalamus. And this is important for the regulation of energy and glucose homeostasis. So we are still actively working on this, and I'm going to end here because it's already a long time. I would like to thank all the members of my lab who've been contributing to this work, as well as all our collaborators at the Institute of Metabolic Science and in Cambridge, and also other collaborators across the world, and of course, all my fundings. Thank you for your attention.
Thank you very much, Dr. Blouet. It was a really fantastic talk. Really spanned over a lot of really interesting areas. We have a couple of questions, and yeah, I'll also add one of my own afterwards. So one question from Sachin here on a part earlier in your talk about the leucine sensing neurons. So they asked, are these responses specific to leucine, or do these neurons respond to other amino acids as well? I think this was in the context of the
in the culture.
Neuronal culture.
So unfortunately, the neurons don't stay very healthy for a very long time. So so we can't really test them, the same neurons with many different signals. And because we wanted really to have the KCL positive control at the end, so what we know is that no neurons respond to valine. So valine is another branched-chain amino acid that does not regulate food intake, but activates many pathways common to leucine, but valine is not detected in the same manner.
Mm-hmm. Okay, and then a question here from Ibrahim on the last part of the talk. So Ibrahim asks, are oligodendrocytes modulated bidirectionally by fasting and refeeding? For example, is there any apoptosis of oligodendrocytes after the transition from fully fed to fasting?
So we haven't been able to see any apoptotic event, and I think it's very difficult to capture apoptosis with such a small population. But we still think oligodendrocytes are modulated bidirectionally. And we know that fasting particularly modulates the progenitors that become less proliferative and differentiate less. So it's not just we feeding, it's both things. And we've looked at the respective effects of fasting and we feeding in details.
Mm-hmm. Okay. And then I had a question about again, about the first part of your talk on leucine sensing neurons. So I was interested in the idea that you presented that there might be different populations of protein or amino acid sensing neurons in different parts of the brain, like in the medial-basal hypothalamus and the NTS. So I remember that you presented data showing that the NTS neurons seem to regulate meal initiation through their projections to the DMH. I was wondering, do they also regulate meal size? And like similarly for the neurons or the CACNA1G mechanism that you're looking at in the hypothalamus, does that seem to be specific to particular parts of feeding microstructure?
Right. Yeah. Carcitonin receptor neurons also regulate meal size. And maybe via different projections. We haven't really characterized that. In the hypothalamus, CACNA1G neurons do not modulate meal initiation, but we've shown that RGRP neurons separately, some work I didn't show that RGRP neurons are leucine sensing and are responsible for the meal initiation aspect of the response.
Mm-hmm. Mm-hmm. Okay. So the question here from Carlos Fibaido, who says, very cool work. I was wondering if calcitonin receptor and CACNA1G are sensing amino acid changes when animals eat protein, i.e. post-ingestive assessment of amino acids, to validate if it is the food that they need, or if they're reporting the protein status of the animal. So kind of, I guess, if they're kind of reinforcing ingestion of a protein-rich food or suppressing ingestion after.
So that's an interesting one, and it's not necessarily straightforward to address experimentally. But what we've done is use this force trap mouse line that with a complicated paradigm, you can label neurons that are responsive to a specific status or to a specific stimulation. And so we've used that to sort of get the central signature of low or high protein status to see what was activated and if there was anything activated chronically. And we didn't find much, and it was quite disappointing. But one thing that we showed that was activated was calcitonin receptor neurons in the NTS, and that's not something that we've published yet. We're still working on it, but we have some evidence that it is the case, that they also may represent the status of the animal.
Mm-hmm. Mm-hmm. Cool. And then another question here from Nevetita. Sorry about that. Do you think that oligodendrocytes in the median eminence regulate neuronal plasticity in response to changes in metabolism?
We don't have evidence that they do.
Yeah, I suppose.
Or maybe indirectly, maybe, but.
Right, through regulating perineural nets. I guess there's some evidence that perineural nets might regulate.
Yeah. Right, so it's indirect. I think it's indirect either via their action on the extracellular matrix or via their action on the permeability barrier, independently of PNNs. But that's all.
Mm-hmm. Mm-hmm. Great. Okay, so that's all of the questions for now. Of course, if anyone wants to discuss further with Dr. Blouet, you're very welcome to join the Meet the Speaker session, as I mentioned at the beginning. The link is in the chat on the right. If you enjoyed this talk, there's a few other things you might be interested in. In the beginning of the talk, Dr. Blouet mentioned the work of Steve Simpson. He presented in our seminar series last year, and that talk's available on our YouTube channel, so you can go check that out. In 2 weeks, Dr. Blouet's colleague, Professor Stephen Rahili, from the Institute of Metabolic Science, will be presenting here as well. And he'll be presenting their work on the genetics of obesity and food intake. And next week, we'll be back to neuroimmunology with Beth Stevens here in Harvard Medical School. So we hope to see you at some of those future talks. I'd like to, again, thank Dr. Blouet for presenting really fantastic talk. And yeah, we'll see you in a few minutes in the Meet the Speaker session.